Our next speaker is Vilu. After a couple of talks looking at the vertebrate system, we've moved back to Vilu's talk, where he's going to, again, I think use C. elegans as a model to look at glutamate receptors and I think, delivery as well as plasticity.
I have this. You can all hear me. Well, I want to thank Sitabra and Srinivasa for the invitation to come to Chennai and present our research. So this is a view of one of the national parks in Utah. I'm at the University of Utah. So I wish this was the view from my window, but it's not far away. It's a few hours south of our lab. So my lab is ultimately very interested in understanding a feature found in essentially all nervous systems. And nervous systems can learn, they're plastic, and with plasticity often comes stable changes called memory. The importance of memory in the face of the change of our life and the change of the nervous system itself, cells growing, cells dying, new connections being made, is truly remarkable. And the importance of memory, I think we can all appreciate, and I like the way Tennessee Williams put it in a play called The Milk Train Doesn't Stop Here Anymore. Life is all memory, except for the one present moment that goes by you so quick you hardly catch it going. And if you think about it, we're all just memory machines, right? Everything we do is based often on past experience and the memory of that past experience. Now, learning and memory are not unique to humans. In essentially any animal with a nervous system, it's been demonstrated that they can learn, they can remember. From seals to mice, to honeybees, to flies, and as you'll see, even extremely simple organisms like worms. Now, many people, many scientists over the last 100 years have tackled the question of memory. And I think there's three large schools. The top-down school, looking at organization of the nervous system, especially regional specialization, to find out what's important for memory. The circuit neurobiologists, well represented here, along with modelers, trying to understand what changes in the circuit, because the circuit's really what makes neurons so fantastic is a single neuron's not really that interesting, but put two together and they can compute something. And then there are people who study the connections between neurons, and these connections are called synapses. And this is where my talk will be focused, the point of contact between synapses. And the reason for this focus is I told you that learning and memory can take place in animals as simple as fly to as complicated as us. And our nervous system may have 100 billion neurons. A worm's nervous system can have 300. So clearly, the evolutionary underpinning of memory is not number of neurons or complexity of connections. Okay? Memory occurred hundreds and hundreds of millions of years ago as a function of nervous systems. And the most evolutionary conserved part of a nervous system is the synapse. And I think one of the real astounding discoveries in the last 30 years of molecular biology and sequencing is that when you look at the gene products in a synapse, from Drosophila to worm to mice to us, they're pretty much evolutionarily conserved. So if we're trying to understand what the molecular underpinnings of memory are, maybe we can exploit this evolutionary conservation. So today's talk will be in three parts. First, I'm going to tell you about published work on the parts of a synapse. And the focus of the parts will be glutamate receptors. Now, glutamate receptors are evolutionary conserved. They're found at essentially all excitatory synapses in all organisms looked at. So we'll understand how those receptors are put there and how they function. Oops. Then I'll move on to the delivery of these components, and finally, their role in plasticity. Now, I mentioned that glutamate receptors are evolutionary conserved. They're found in all nervous systems, and they're classified in three major groups called AMPA, kainate, and NMDA receptors. And these are the drugs that preferentially activate these classes of receptors. But the receptors themselves all have a very conserved architecture. This is a top-down view. It's a tetramer of 4 different protein components, protein subunits that surround a central pore. When glutamate, the neurotransmitter, binds, there's a conformational change, a hole opens and ions can flow through. And if you record from a cell in which you've activated these glutamate receptors, you see there's no current, then you spritz on glutamate, and you see, boom, there's an inward current, and then that current, in the continued presence of the glutamate, desensitizes. And that's because the receptor now has a conformational change, even when bound to glutamate, that closes the pore. So you can think of this maybe as a fail-safe mechanism, and I'll try and make the argument that it's actually a way of changing the bandwidth of the synaptic transfer function. So this is the top-down view, and here's the side view. You can see here that the receptor binds, that there's glutamate binding sites on the receptor shown here, and that causes a conformational change in the middle, allowing ions to flow through. Now, in the last 20 years, there's been an enormous change in our thinking of what plasticity means at synapses. So if you buy my argument that changes in synaptic strength, how one neuron talks to the other neuron, is an integral part of learning and memory, then the next question is, well, what's changing? And of course, you focus on that which you have in hand. And what's in hand is we know that there are glutamate receptors at these synapses. And I would say until the 1990s, the predominant view was that signaling cascades caused changes in the status of those receptors due to phosphorylation and other post-translational modifications that's changed their receptor properties, making them either stronger or maybe weaker. Okay? Now, that's not the way people think about it as much anymore because cell biology has entered the picture. We now know, if you look at, for example, plasticity, and the most famous example of that are the LTP, or long-term potentiation protocols, or long-term depression protocols, that with these protocols you can change synaptic strength. You can make the strength stronger, and the thought is one way you make it stronger is you put more receptors into the membrane. So think of the receptors as amplifiers, and you're putting in more amplifiers, and of course the strength is going to be stronger. And there are other protocols that weaken synapses. These are called like LTD. And with that, you have a removal of receptors. So the notion now is that there's dynamic movement with experience, changing the number of receptors at the synapse. And we'd very much like to understand how that takes place. So, given the importance of these receptors to not just synaptic function, which of course they're going to be important for synaptic function, but that their numbers and properties might be modified with experience, it's obviously of immense importance to understand really from a soup to nuts point of view how these receptors are synthesized, trafficked, targeted so they go to this synapse and they don't go to the wrong place, how they're inserted into the membrane by experience, and how they function. Okay? So how can we figure this out in a kind of systematic way to really understand how do you build a synapse? So what I'm going to tell you today in the first half of the story of my seminar is a synapse in C. elegans and the genetic studies that have uncovered the role of different gene products to synaptic function. So at the beginning of our work, which is over a decade now, I would think that the majority of the people in the field thought that glutamate receptors were standalone molecules. And I'm going to provide evidence to you that they are not. They are part of a very complicated signaling machine, and that this signaling machine provides synaptic complexity. So, first I'm going to tell you about our genetic strategy. So I will try and make you all geneticists after this talk. Second, I'll tell you about one example called SOL1, just so you understand how this genetic strategy takes place, and that's already been published. Then I'll tell you a little bit more about other components of the receptor complex, including a paper that just came out in Neuron last month. So, if you want to understand the parts of synaptic communication, and especially how receptors are modified with experience, which we think underlies learning and memory, you can take advantage of the simplest system that has synapses, because I told you synapses are evolutionarily conserved. And we've taken advantage of C. elegans, the simple nematode that's been described very nicely in the talks earlier this morning. And I'm just going to reinforce some of the points raised by May and Sandia and Kathy. This animal has 302 neurons and in aggregate maybe 6,000 or 7,000 synapses. So let's put that into perspective, and especially when you see what the worm can do. This has the same number of synapses as one CA1 hippocampal pyramidal cell, or one GP neuron, roughly. Okay? So the entire animal has about the same synaptic complexity as one neuron in our brains. It's transparent, and this you'll see we exploit as Sandia did earlier today. And it's also amenable to electrophysiological analysis. So I show you here a subset of those 302 neurons, including the interneurons that May told you about that control the direction of the worm, whether it goes forward or backwards. And we know that there are sensory neurons that release glutamate. It depends on this particular protein called EAT4, which packages glutamate into the presynaptic vesicles. Then these make synaptic connections onto the interneurons, which tell the worm to go either forward or backwards. So this simple circuit gives us a fantastic system to try and understand what gene products are important for synaptic function. I told you that that circuit is required for backward movement. So a geneticist would say, okay, I will randomly mutagenize these worms. They're only a millimeter long. On a plate, you can put on 2,000 very easily. And I will look for those gene products when disrupted mess up the avoidance backing behavior. So if this synapse, which uses glutamate receptors, is important for the worm to go backwards, and I mutagenize a key component, it won't go backwards. So that's a theory. And if I had enough hands and enough willingness, you could actually do these kind of screens, but they're actually very difficult to do because they're boring and labor intensive. And so 2 students in my lab devised a much better way of doing the screen. And what they did is the screen in a sensitized genetic background, which I'll tell you about. So this is a wild-type worm, a normal worm. Most of its time, when it's off food, it explores and it goes forward, as was described earlier. And periodically, it'll back up, and with that backing up, it reorients and goes off in a new direction, foraging. Pretty amazing, only 302 neurons. Look at that. I have 100 billion, and I can't go that smoothly. So what Penny Brockie and Yi Zheng did in my lab was to engineer into the glutamate receptor gain-of-function mutation and put it back into the worm. So this worm is no different than the worm I showed you previously. It has a single amino acid change in one receptor in its genome. And perhaps you can notice that this worm doesn't look wild-type. It doesn't look normal. It moves sinusoidally very well, but it goes back and forth, back and forth, back and forth. And you might think, what a trivial observation, who cares? But to a geneticist, this is an extremely attractive tool. Because what you can do now is to use very powerful population-based techniques to look for gene products when mutated that will disrupt that pattern of behavior. So imagine you, and they just go back and forth, back and forth. And because of that, that takes them forever. They can detect the food. They yearn the food. They want to be at the food. But they are compelled to take one step backward following each step forward. So the point is that there's an enormous difference in the behavior of these worms. Now here's where the genetic experiment comes in. You mutagenize with a typical mutagenesis protocol. Mutagenizing means that you disrupt the DNA that encodes proteins. And you look for suppressors. And for this strategy to work, you need a control to convince yourself that the strategy will work. So the control here is to knock out the normal glutamate receptor. In other words, if I want to look for modifiers of the gain-of-function glutamate receptor, imagine I have a modifier that just knocks out expression altogether. The protein's never made. I have a control for that. And that control is to find the strain that doesn't have the glutamate receptor, and that's this. So the yellow line, yellow-greenish line, shows the control. So if I find a mutation that prevents this from working, it should look like that. Okay, that's a screen, and I can do it now in industrial quantities and look for rare mutants that make it to the food. And when I did that, well, when we did that, Penny and Yi Zhang and others, we found our first suppressor. Notice it's behaving much better than the starting strain, but it's not normal, which is what you'd expect. Glutamate receptors probably are important. So what is this gene? Well, using conventional cloning techniques in C. elegans, we can identify the gene itself. And this was called SOL1 for suppressor of Lurcher. And it's an interesting protein. Many proteins like this are found in our brains. It has a single transmembrane domain with 4 protein-protein interaction motifs called cub domains. And we found many different alleles. That means when we did the screen, we kept getting this gene, so showing it was very important. Now, a suppressor could be specific to the gain-of-function mutant. We put in a weird glutamate receptor. Is this specific to that? So we just do the standard thing. We get rid of that weird gain-of-function background, and we just look at the mutant itself. If it's really important for glutamatergic neurotransmission, it should look like a glutamate receptor mutant. And so this is an assay for the avoidance response, and here you see wild-type worms avoid very well. If you don't have glutamate receptors, you don't avoid very well. If you don't have SOL1, you also don't avoid very well. And we can put that back just in those neurons and show that now the worms avoid very well. So by behavioral criteria, SOL1 is important for synaptic function, mediated by glutamate. So where's SOL1 acting? With a beautiful genetic screen like this, we should eventually get the entire signaling pathway from nucleus to synapse. But of course, we want to know where in that large signaling pathway things work. So in a SOL1 mutant, we find that the glutamate receptors, these AMPA receptors that I discussed, they're actually at the synapses and at the cell surface. So it looks like there's no signaling, but the receptors are there. So to figure out what's going on, we turn to electrophysiological analysis. And the enabling technology here is, one, the worm itself. It's clear. It's transparent. So you can make a transgenic worm that expresses GFP, a fluorescent protein, in specific neurons. You can glue the worm down. You can then dissect carefully the cuticle to expose the neurons. There are the neurons 1, 2, 3, 4. And because it only has 302 neurons, you can always go to the exact same neuron again using electrophysiology and a pipette, patch pipette, and record from this neuron. So this is the neuron AVA that was discussed by May earlier. And when we record from this neuron, we see, and this is in the wild-type control, and we spritz on glutamate, we can see robust glutamate-gated currents from that neuron, AVA. Now what happens in the SOL1 mutant? Well, when we put on glutamate, we find that the currents are markedly different. They're very diminished. Essentially, there's no apparent inward current at minus 60 millivolts. But at plus 40 millivolts, we still see that there is some current remaining. How does this compare to a lack of the glutamate receptor itself? So here's the GLUT1 mutant. It has no glutamate receptor. And look at that. The electrophysiological signatures are almost identical. So I told you that SOL1 looks just like a GLUT1 mutant in terms of behavior. And in terms of electrophysiology, they also look identical. And you can rescue that by putting a wild-type copy of SOL1 into these neurons. Now, this particular result was extremely pleasing because electrophysiologists in the vertebrate field always wonder, well, you can come up with any model in the worm because you can't actually figure anything out because you can't record things. Well, we can record the currents here. And look at this electrophysiological signature. I told you that not all the glutamate-gated current is gone. There's another component. And for those of you in the field, you'll recognize that there's an outward component that remains here. And this is the NMDA component. So the NMDA component, a different flavor of glutamate receptors, is untouched. This is very important. It says that SOL1 is not just some non-specific thing that's messing up synapses in general or the health of the cell in general. It's saying it's very specific and it's tied to the AMPA receptors that we're particularly interested in terms of plasticity. So, does SOL1 regulate glutamate receptor activation or desensitization or binding or what? I mean, the receptors are there, they don't seem to work. Why? Now, we know from recent structural studies that the isolated glutamate receptor can bind glutamate just fine. So there's been X-ray crystallography studies showing that works. So logically, we can think either glutamate no longer turns the receptor on in the SOL1 mutant, or in the SOL1 mutant, the receptor is on, but it turns off so quickly we don't even see it. So how do we distinguish that? I showed you if I puffed on glutamate, and I didn't tell you I puffed it on, but when those experiments, when I showed you the glutamate currents, I just puffed on glutamate. So puffing on glutamate is actually a rather slow process. What you want to do is have a step function where you just turn on glutamate. It goes from 0 to 100% instantaneously, kind of like this. Boom. And if you did that, you would see the true kinetics of the receptor. Okay? But if you can't do that kind of experiment and you dribble on the glutamate slowly, like here, so what you're doing now is you're only activating a subset of the neurons, of the receptors on the cell at a time. They desensitize. And so this current, the magnitude, the area under the curve of this current is exactly the same as the area under the curve here. But you can imagine as you put the drug on slower and slower and slower, this line could look flat. Okay? It's a very important concept. Everyone follow? Okay, so with that, we had to be able to apply drug to the neurons in the worm faster than roughly a millisecond, because that's the kinetics of the channel. And the way we did this is use a piezoelectric device to drive a double-barrel pipette, and that kind of works like this. Very quickly, it switches in only a few hundred microseconds from one stream to the other. And when we did this, we looked at neurons, and let me just tell you, this is the glutamate receptor. This is another protein that's required for function, which I won't tell you about right now, and this is SOL1. And when you rapidly apply glutamate, you see very quickly a current turns on in only a few hundred microseconds, and then it desensitizes. See, the glutamate's still there, but the current is getting smaller and smaller and gets to a plateau level. Now you wait 300 milliseconds, and you try it again, and you see that only about half the current has recovered. Now, if you do the same pairing of experiment with many seconds in between, you see that the initial curve always recovers, and then with time, if you wait about a second and a half between pulses, you see full recovery. Okay? Two important points. The receptors desensitize very quickly, and the receptors take some time to recover from desensitization. So now what happens if we don't have SOL1? Well, how many people think it's activation? How many people think it's desensitization? Well, I wouldn't have gone through the whole desensitization argument if it wasn't desensitization, but that's what it is. And this is a very beautiful series of experiments. You leave out SOL1, you see that the channel turns on just fine, but it almost instantly desensitizes, and it takes quite a while to recover from that desensitization. Now, for the modelers amongst you, you can think, how could you change the bandwidth of a synapse and the function of a synapse by changing the rate of desensitization? But I'm not going to go there, because that's where I'm very weak. I'll move on and tell you that we use this approach to show that really glutamate receptors, AMPA receptors, are signaling complexes. The glutamate receptor itself is the central part. That's where the pore is. But we've identified now two classes of proteins called stargazins, or TARPs, and SOL1. And so this is all, these are all experiments demonstrating necessity of these gene products for synaptic function. So it was also nice to test one's model by showing sufficiency. And I love this quote by Richard Feynman: "What I cannot create, I do not understand." And so as we get the parts, we'd like to figure out how they are put together and how they work. And so we take these gene products, and we express them in Xenopus oocytes. So we think glutamate receptors are important for glutamate-gated current, as everyone would agree. But when we put this into Xenopus oocyte and throw on glutamate, we see no current. Then our genetic experiment said, yes, but you need Sol1. It's a critical component. So we do that, and we put GLUR1 and Sol1 into the Xenopus oocyte, and there's still no current. And then we also identified another protein essential called STG1, and we put that into oocytes, and we saw no current. So we were getting dismayed, but when we put all three together, you could see that there's a very large, robust glutamate-gated current, really demonstrating that there's a signaling complex. It's not just a channel. It's a channel in an environment. Okay? And we'd like to understand more about that environment. Now, one thing we noticed is that this current still doesn't look like the current in the neurons themselves. So we did yet another screen, and we found, with the same strategy, a little more sophisticated variant of it, yet another cub domain protein called Sol2. And so this protein has homologs. So this is compared to our Sol1 protein. And Sol2 looks like other proteins found in mammals called NETO1 and NETO2 that have independently been shown to be important for NMDA receptor function and AMPA receptor function. So in a Sol2 mutant, you see also that the currents are very diminished, and you can rescue that behavior. You can ask the question, since Sol1 and Sol2, if you knock either one of them out, the behavioral phenotype looks the same and the current reduction looks the same. Do they associate? And we did a trick called bifluorescence complementation. You take a protein A that you think matches to protein B, and you put 1/2 of YFP on it. And this doesn't work, ever. And on the other protein, you put another 1/2 of YFP, and that doesn't work. But if A and B truly come together, this protein gets reconstituted, and you can see the GFP signal, or the YFP signal. So we did that. Here's the 2 halves of Sol1 and Sol2, here's Sol1 and Sol2, each tagged with 1/2 of a YFP, and you can see these nice puncta along the process, the same kind of process that Cynthia showed earlier, and we believe these are points of synaptic contact. So these go to the synapses. And if you label with another fluorophore cherry GLUR1, the glutamate receptor, you see they also go to the synapse. And in fact, they all co-localize, giving some cell biological credence to our genetic arguments that these are all part of a signaling complex. And Sol2 has the same type of effect on receptor desensitization. This just shows, again, rapid perfusion studies showing the markedly different desensitization rates. So what have I shown you in the first part of the talk? What I've tried to convince you of is genetics, using this principle that you can, by scanning large populations, find informative mutations for a process of interest. We've uncovered that glutamate receptor signaling is really through a signaling complex. And so the questions we're interested in now is how is the complex assembled and targeted and regulated? We think these could provide very interesting targets for drug discovery efforts. Most drugs that are effective in treating neurological psychiatric disorders are at the level of the synapse. There are other flavors of glutamate receptors called kainate NMDA receptors. We believe, based on this model, that there must be auxiliary subunits for those receptors as well. And of course, we think these could be extremely interesting as possible roles in synaptic plasticity, and I'll get back to this later. So let me tell you now, if we believe these signaling complexes are important as I suggest, and I think you'll all agree with me, then they have to be put in the right place for 2 reasons. A, you have to signal at specific synapses. B, if you put these receptors in the wrong place, not only will you mess up signaling, you'll mess up network properties, and you can provoke neuronal death through something called excitotoxicity. So how do receptors that are mostly synthesized in the cell body get delivered to distant synapses, which can be hundreds of microns away? And there's 3 major models. One is that there are molecular motors. And surprisingly, in the entire field of AMPA receptor biology, where there's probably 10,000 papers, there's only a handful, even fewer, showing the involvement of motors in transport of glutamate receptors. And in fact, there are people who believe that that's not how it works. Roger Nichols' group thinks it's by diffusion through the plane of the membrane and then trapping at synapses. And I would say that there's a major component of current thinking that believes that the messenger RNA itself is what gets located to synapses and that there's local translation. So how can we start distinguishing between these models, which are very different? And that's the beauty of biology. So you can have models that differ in their predictions by 2 orders of magnitude. That's what you want to write your grant for and try and solve. So we take advantage again of the worm. This shows the neuron AVA. And the green dots are glutamate receptors tagged with GFP, and they localize in these puncta, which Josh Kaplan and Chris Rongo and others have shown are almost certainly points of synaptic contact. So these are accumulations of receptor at synapses. And if you blow up this region right around here, you can see much more clearly that there are very stable, bright puncta of receptors. So how do they get there? Well, one approach is to do in vivo imaging. So this is in a live worm. It's just on a microscope. It's happy as a clam. That's an American expression. And to clean this up a bit, we photo-bleached the entire process in this region. And so we have now a very nice black background, so we can really see movement of receptors. And then we start taking a streaming confocal movie to see what's going on. And when we do this, we see, oh my God, it's like a super highway. You can see these glutamate receptors going, they kind of stop, they jitter. Look at this one, it's going to stop right there, right where that synapse was. Okay? The other thing you can notice is that the movement goes in both directions. Okay? So this would be anterograde and this is retrograde, both directions. The movement is dependent on ATP. You can poison it with azide, cyanide. The movement is dependent on microtubules, so almost certainly it's due to motors. And the movement is around 1.6 microns per second, consistent with motor-driven movement, as described earlier today by Cynthia. So you can take these movies, and here's, again, the same. Here are the synapses, and then we bleach them. And now you can very clearly, in this chymograph, which are just these images stacked on top of each other, you can see it, for example, there's a vesicle moving here, carrying glutamate receptors. Boom, it stops. And then it stays in this one position. And 20 minutes later, it's still in that one position. If you look through, let's say, this window of interest here, B and A, you can see this thing goes through, and it lands, and you can see these quantal increases in fluorescence. But you can also see it coming from the opposite direction. So there's a track coming here, and its quantal input is right there. So the take-home message is that receptors are going in both directions and they can land on existing synapses. And we can look at that more carefully by, here are the synapses, and then we can fit each green dot is the location of those synapses. And then we can ask, where do the stoppage events occur? Because I was very, provocative saying it looks like they stop at existing synapses. And in fact, you can see that they do stop mostly near existing synapses, and you can quantify that. It looks like there's an exponential distribution. The most likely place to stop at the synapse, and then it falls off on either side. So let's look at that a little bit more. I'm suggesting that the delivery of glutamate receptors is via the motor. So again, we can bleach the whole thing, and we can bleach the signal. And here we use a very different kind of signal. We use 2 signals. So M-cherry, that's the red signal, and a pH-sensitive version of GFP called SEP. So if it's in a vesicle being transported, like here, it's quenched by the acidic pH of the vesicle. Once it pops into the surface membrane, it's unquenched and it can become green. And so you can see, and maybe you can't, but there's a track coming in here. There's a vesicle being delivered, and then it stops, and it stays here. Stays here, and boom. At this point, it became accessible and is at the synapse itself. So I think these data really suggest that motors deliver receptors to synapses. What might the motors be? So we did a genetic screen looking for what the responsible motor might be, and we found that it's kinesin 1. And the heavy chain of kinesin 1 is called UNC116. It doesn't matter, but it is the motor. And so you can see these chimegraphs in wild-type worms. This is the glutamate receptors traveling in both directions. And here's an allele of UNC116 called E2310. Doesn't matter. It just means it's a crippled motor. It doesn't work very well. You can see just by inspection at the back of the room that the tracks don't look quite right. And here's a stronger allele, and now it really looks like it doesn't work well. Here's even the stronger allele, and there's almost no movement whatsoever. Here is a dominant negative mutation that we introduced only in this neuron to squash the activity of the motor. And now you see no movement. And here is using an RNAi approach to get rid of the endogenous motor, no movement. So what I'm telling you is that you need UNC116, you need kinesin 1 to have movement. If you don't have it, no movement. Okay? Now, for those of you who are aficionados of this, you'd say, oh, but you told me that the vesicles move in both directions. And I know that kinesins are plus-end motors. And so I don't have time to show you this, but I've shown that in this AVA process, the microtubules are actually in both orientations. So they go in both directions. So the motor can go in either direction as well. Now, this could be a direct effect or an indirect effect of the motor. Okay? And so to look to see if it's direct effect, what we did is we labeled the receptor with M cherry, so it's red, and we labeled the motor with GFP. And you can see in dual streaming movies that the motor and the receptor always move together. Okay, so is kinesin 1 required for delivery of the receptors? What we did here is we just did an experiment now in the adult worm. Okay? We're not looking at mutants anymore. Well, we are looking at mutants, but we're looking at acute perturbations. Let me explain. So here's the distal half of the worm. So the process itself is about 1 millimeter. 1 millimeter, 1,000 microns. For something to diffuse that long would take days. Okay? When we photo bleach, that means we just get rid of, we bleach all of the GFP signal in the distal half of the worm. And then we look in these regions, S1, S2, S3. So here's the wild type. There, the puncta. The next line shows bleaching. There's nothing here. So this is called a FRAP, or fluorescence recovery after photo bleaching experiment. And now you can see that 4 hours later, in S1 you see signal, it comes back. S2 you see signal. S3 you see pretty much full recovery in 4 hours. But in an UNC116 mutant, even though you can see puncta here, you see essentially no recovery. Now, unfortunately, the lights aren't good enough for you to really see that well, so I'll show it to you another way. This is just a line scan showing the intensity of puncta along the entire distribution here. So that's before we bleach, and you see that the signal all goes away. And then 4 hours later, you see the signal comes back in wild type. But in UNC116 that has no motor, the signal doesn't come back. Now, you can imagine that the signal comes back because of motor-dependent delivery, which is what I'm arguing, or that there's actually a strong local synthesis. This has nothing to really do with the essential event. So you can block new protein translation with a drug called cyclohexamide. And so when we do the same kind of bleach experiment, so this is before, we bleach, and then we look at recovery, we see very nice recovery. If we do this in the presence of cyclohexamide, so you make no new proteins, you still see completely normal recovery. So this indicates, I set up 3 models for you: diffusion, local translation, or motors. It looks like it's motors, and it looks like it's kinesin 1. Now, if you look at these kinesin mutants, the different alleles, you see, in fact, that there are receptors out there. And I just told you that you don't get very good receptors out there. in the absence of the motor. And this is a key thing that many geneticists don't actually appreciate. We look at worms that are 4 days old. 96 hours is a long time for proteins to diffuse. And so it looks like in that time period, receptors can diffuse out there, and they are trapped by synapses, at synapses. And in fact, they look like there's way more in an UNC mutant than in wild type. Like they diffuse out there, but they can't be removed. And so we tested that hypothesis to see if the motors are also important for removing receptors. And we used a type of fluorophore, a variant of GFP, that you turn on with UV illumination. So initially, there's no signal. And then, boom, we turn them all on. And then we ask, how long does it take for it to go away? The only way it's going to go away is if something removes it. And you can see in a wild-type worm, in 4 hours, most of the receptors are removed. But in UNC116 mutants, the removal rate is very much slower. And you can it's quantified here. So what I try to argue now is there is a receptor complex. We've shown what the components are. And we've shown that at least one part of this receptor complex needs motors to get out there. And since I started out to talk about synaptic plasticity, learning, and memory, and saying that the exchange of receptors is really important, the next natural question to ask is, how important are motors for learning and memory and plasticity? And so that's what I want to finish my talk with. This is all unpublished, hopefully soon to be published. And I'm going to remind you, put everyone here that has, we all have a common interest in how neural networks work. And one of the best models of learning and memory is called long-term potentiation. And the idea is that independent synapses can be potentiated, right? Part of the complexity and power of our nervous system is that we have independent regulation and strengthening of synapses. Now, how do you independently regulate this? This is a very thorny problem because if the receptors are made here, how do you know which synapse to potentiate and how? And so a model that's been developed over the past 10 years, not the only model, is something called synaptic tag and capture. And the idea kind of goes like this. If you use LTP, for example, to tetanize or stimulate one synapse, make it learn, that there is a tag, there's some kind of change, a molecular, perhaps structural change that says this synapse is different. Okay? That's a tag. The other part of the hypothesis is the capture hypothesis. And the thought is that this strong stimulus somehow causes local protein translation, and that local protein translation is captured by the tag. Okay? Synaptic tag and capture. So can we test that model? Maybe extend these kind of this thinking using C. elegans. And so what we've taken advantage of is this light-gated channel called channel rhodopsin. We put it into the neurons that release glutamate, and we turn on blue light. And you can see every time we turn on blue light, the worm goes backward, as predicted and shown early by Meijin, right? That you can stimulate neurons using channel rhodopsin. Okay? So it goes backwards. That's nice. We can show that, in fact, it's a very, it's light dependent. So the probability response goes up with how much you depolarize the neuron. And it's dependent on the two players we absolutely know are important for synaptic neurotransmission, the presynaptic vesicular uptake protein, EAT4, and the postsynaptic receptor. Okay? So if you don't have the presynaptic uptake protein, you have no glutamatergic transmission, you get no backing response. If you lack the glutamate receptor, you have a huge decrease in the probability of response. Remember, there are other glutamate receptors there as well, which I've glossed over and not told you about. So this is very nice. And you can show that you can use light as a training protocol. So we can expose worms for an hour to blinking blue light, our own version of long-term potentiation or long-term facilitation, and ask what happens. And what you see is on the top, we show that glutamate receptors, either untrained or trained, and when tagged with GFP, you don't see much change. But when tagged with SEP, which shows the number of receptors at the surface, you see there's a dramatic change in the trained worms. Okay? So we've shown now plasticity. Depending on a training protocol, we can change the number of receptors at a synapse, and we think the number of receptors at a synapse is going to be part and parcel of learning and memory. Well, we know quite a bit from vertebrate work about molecular gene products that are known to be important for this process. And we know that NMDA receptors are important. They're expressed in these neurons as well, and you don't see the change in an NMDA receptor knockout. UNC43 is the famous CAM kinase 2. We don't see the change in a CAM kinase 2 knockout. If we take a double mutant between CAM kinase 2 and NMDA receptors, we can ask the question, do they function in the same signaling pathway? If they function in the same pathway, the double mutant should be no worse than the single mutants, and that's what we find. So this is not totally unexpected, but what is unexpected is that we are linking NMDA receptors and UNC43, the CAM kinase, to the motor protein, because we don't see changes in the motor protein mutants either. So our model is when you activate a synapse, you're putting out a tag, and that tag is telling the motor to stop and deliver its cargo of receptor. And that is what's required for plasticity. And you can even think that this is a sustaining way to have receptors be strong for a long time. It could be a positive feedback loop, right? If you stop, you deliver more cargo, you have more receptors, you have more of a signal, you maintain that stop signal. That's our model. So can we look at that in real time? And so here we look at these synapses in the presence of retinal. I didn't tell you retinal is an essential cofactor for channel rhodopsin to work. And we turn the light on for periods and off for periods, on, off. And here are the chymographic tracks. And when we see, in the absence of retinal, we see very little effect of light. But when we put retinal in there, you can see that these tracks become very different in the presence of light. The average run length decreases. So you can think there's a train moving with its cargo, and now there's a new sign saying stop. And since we're activating all the synapses, the average duration of the train moving its cargo is far shorter. Does everyone follow? Okay. So, optogenetic tagging and capture, our model is the following. That receptors, you can think of synapses as, in America, 7-Eleven stores. Okay? The 7-Eleven store is all throughout the highway, and it provides goods. So it's like the synapse. But it needs trucks all the time to deliver and remove fresh bread and take out the old eggs and all that. And the only way you can tell us whether a synapse needs new supplies is to put a tag. Okay? So the tag is from the activity. It goes through receptors, the NMDA receptors, the GLUR1 receptors. In some process we don't understand, UNC43 is critical and sets a tag allowing for drop-off and delivery of cargo. And so we think this is the way not only that plasticity might be initiated in the nervous system, but may also be sustained. So I want to finish with a model. This is, in the old dusty towns of the American West, trains didn't have time to stop at stations. It was too inefficient. You have this huge inertial load. You have to start it all up again. So they had what's called a mail hook. So there's the station master, and he puts the mail on a hook. And you can see that there's a train coming. There it is. So the train is driven by UNC116, kinesin 1. And it's coming along. And it's not going to stop. Oh, but there's a mail hook out. Okay, because the mail hook is out, it doesn't even have to stop. And what it's going to do is go by. The hook triggers things, and you see first the new receptors being thrown out. There they come, and the old ones being removed. Okay? And so this is the way I think synapses work. Learning and memory occurs, happiness, bliss, all that is due to motors delivering things and taking them away. So I want to pay particular attention, you should pay particular attention to the postdoc Frederick Hernley and a grad student Dane Maxfield that did the majority of the second part of the talk. And what I didn't mention was all the work on Sol2 was by Ray Wang, an extremely talented grad student who recently graduated. And with that, and thanks to my funding agencies, I'll take questions. Yes? Oh.
I have a couple of questions. That was terrific. How do you think the fact that C. elegans...
I'm trying to focus where the voice is coming. Oh, thank you.
How do you think C. elegans doesn't... The sensory neurons aren't producing action potentials. We're getting graded release of neurotransmitter. And what you talked about was the difference in when transmitters released very rapidly. Do you think that the sensory neurons are releasing it very rapidly, or is there some dynamic as they're working in vivo?
Well, the question was about the distinction between, yikes, still works. The distinction between action potentials and graded potentials. And I guess from my perspective, from an evolutionary point of view, action potentials are very recent. They're good for long-range transmission between one module of neurons to another. Okay? But just because something's graded doesn't mean it's not fast. Okay? So you can be near threshold, let's say minus 40 millivolts, and very quickly, within milliseconds, calcium channels open up, and you get influx of calcium, and in less than a millisecond, you get release of neurotransmitters. So I don't think that has much to do with it. It makes maybe the interpretation of channel adoption experiments a little harder because we don't have the way of measuring frequency of action potentials. But the concept is still the same. You're quickly changing the probability of release.
Okay. And when you photo bleached and used cyclohexamide, and you still got glutamate receptors, where are they coming from? Are they spreading out along the process? Because it's not going to be making new ones.
Well, so the question is, where do the receptors come from when you are blocking protein translation? And that experiment was to test the hypothesis that local protein translation was important for recovery from photobleaching. So at least 80%, maybe more, is already made in the cell body. And in experiments I didn't show you, we've been able to demonstrate that receptors are actually redistributed between synapses, okay, which has actually incredible implications for homeostatic plasticity. So you can still have a huge, Costco warehouse full of glutamate receptors that can be delivered, and you don't need to make new receptors at the point of the synapse. So that was just to test that one specific hypothesis that the new receptors would come from RNA clustered underneath the synapse itself. Yes?
Thanks for a very nice talk. The last part with the transport of the glutamate receptors that were trapped at the active synapse. Now, that reminds me of Richard Morris. He proposed a similar type of interpretation in vertebrates some years ago. Isn't that right?
Well, you're exposing the fact that when I introduced the synaptic tag and capture, that I said it's been around for 10 years. It was started by Richard Morris.
Yeah, okay. I missed that.
Yeah.
Because I was a little surprised, and I'm not an expert in this molecular signaling complexes, but I teach some of the stuff to undergraduates, and the word scaffolding protein comes up all the time. So I was surprised that you didn't pull that all the complex was hanging together without a scaffold, or did I get that wrong?
Well, no, you didn't get that wrong. What you identified was the fact that what you point out is our genetic experiments found molecules that were important mostly for function. You might ask, why didn't our genetic screens get scaffolding proteins? And, well, in fact, we have lots of mutants we haven't analyzed, and we may have scaffolding proteins in there. But I suspect that the reason we didn't look at those mutants, which I'm sure we have, is because they have pleiotropic effects. These scaffolding proteins are not limited to glutamate receptors. So whatever cartoons you have of PSD95 or whatever, the only thing, it's not that only AMPA receptors stick to them. So if you knock them out, you might expect dramatic changes in behavior. The reason that we picked SOL1 and SOL2, I mean, we studied these and identified them in our screens, is that they most phenocopied the loss of the receptor itself. So, in fact, we think that there are many other gene products, including scaffolding proteins, that we will find in our genetic screens.
Great talk. I have two questions. With the transport of UNC 16, is it just transporting the GLORA1 by itself, or is it transporting the entire complex that local?
So May asks a very fundamental question, which is, I said there's a signaling complex, and you can imagine that the complex is put together already at the level of the ER, and then the whole thing is shipped out, or that it's independently shipped out. And our evidence so far is that it's independently shipped out.
So all of them?
Not all of them, just for SOL1 and GLUR1.
And then do you have any evidence this bigger punctus in UNC16 mutant are inserted in the plasma membrane, not trapped in some kind of like endosomes and stuff?
Yeah, so that's also a good question. So for those of you who didn't hear, May asked, I showed that there are a greater number of receptors at synapses in the UNC116 mutant. This was surprising. There were bigger accumulations. And she asked, is that due to trapping in the subsynaptic, in other words, in the intracellular compartments, or are they actually at the surface? And the answer is both. There's greater numbers at the surface, and there's greater numbers subsynaptically. So it scales. There's just more there, but there are more at the surface as well.
So
Yes.
I like the illustration of the train, and in fact, in following May's question, my understanding of it always was that something like the train, you know, your motor is carrying a whole lot of things. It's a sort of fuzzball, including mRNAs and proteins and other things. And it wasn't clear to me whether you had in mind that one component of this fuzzball would come off at the target synapse, or whether the whole thing would come off and then the target synapse would harvest whatever was needed.
Yeah, that's a very deep, interesting question, which is how do you get cargo specificity? And we are not sure about that. But we do know that there are other gene products, including UNC16, UNC14, KLC2. These are either parts of the motor or cargo adapters that phenocopy this defect. So we know that there is some signal there, but we don't know, you know, the cartoon is it's a beach ball and the receptors are stuck in that beach ball. So the train is certainly carrying that one ball and it drops it off. But what else is in that ball is there's no easy way to figure that out. That's going to just take brute force, seeing what's there by genetics, I think. If you're a biochemist, you'd disagree with me there.
Okay, I wanted to ask a question.
Yeah.
So you show with the optogenetic manipulation as well as with the motor that you have many more receptors on the surface. So how does the behavior of the animal change?
Yeah, I was afraid someone was going to ask that question. So we're currently trying, obviously, we would love to be able to tie together the cell biology, the behavior, and electrophysiology. I mean, this is the holy grail. And remember what we're doing is we're potentiating synapses, and we can look at those individual synapses, but we're potentiating all of them. Every glutamate receptor, every glutamatergic synapse is being potentiated because of the promoter that we use, which is called the E4 promoter. So it's in all of the neurons. And really what you want to do, and so it's hard to predict if you potentiate everything, how will behavior change. Okay? We think it changes, and we're looking using the worm tracker we see quantifiable changes in behavior. What we really want is far more specific promoters to test, for example, are you more likely to recoil to an avoidance stimulus. The problem is those promoters in our hands have not proved powerful enough to drive the optogenetic stimulation. So that's a work in progress. But I think the concept is pretty clear. The concept is you can change the activity of synapses, so you can kind of teach by remote control. And one of the difficulties in the worm work is how to synchronize learning, how to develop behavioral paradigms. This offers perhaps an alternative of using light to do it. Right? So you can now teach lots of worms all at the same time using light. But we're not there yet, but we think we have a path to there.